So welcome to this class. In the previous lecture, I just introduced the frequency distribution analysis, what is called binomial analysis. Then we also covered about the cluster identification, okay, and briefly I talked about the radial distribution maps, correct. So and also I talked about the nearest neighbor distribution, fine. So first nearest neighbor, second nearest neighbor, okay. So based on that, how to identify the clusters in a given data set. Now briefly I will just introduce the effect of detector efficiency. So you know that detector efficiency. So I think long before we just discussed about this efficiency. It depends upon the ions which are collected at the MCP multi-channel plates, correct. And we know that these multi-channel plates, these are the glass tubes, and the whatever area which is not covered in the glass tubes, usually those ions will be reflected back or they will be lost. So there will be a detector efficiency depending upon the design of MC plates. So it will not be 100%, okay. It might be around some instruments they have, it ranges from 40% to 75%, correct, depending upon the design of the instrument. So this detector efficiency will also affect the cluster analysis, will also affect the cluster analysis, okay. So first we will discuss about the nearest neighbor. First we will talk about the kth nearest neighbor, effect of detector efficiency on KNN, okay. So it will affect in two ways. Limited detector efficiency induces, first it will induce a peak shift to the right in KNN nearest neighbor, okay. Why it will shift to the right? So if you have a, then if you have a this particular peak for the nearest neighbor distribution, then usually the peak will shift in the right side, okay, at a larger distances, correct. It means that less atoms, it means that the average distances between the pairs is increased, okay. And the second thing is it usually causes widening of distribution, okay. Why widening of the distribution? Because the limited detection efficiency will actually remove fraction of atoms which are in the nearest neighbor, okay. So these are the two important things which usually affects the detector efficiency affects the during the nearest neighbor distribution, okay. So I will show you a chart where you can see that the solute nearest distance, and this is the frequency, okay. And you can see that the blue color, these are the simulated data. So where the blue color corresponds to 100% detection efficiency, red color 57, and the green color is 37%. Now as the detection efficiency is reducing, correct, from 100 to 57 to 37%. So means out of 100 ions only 37 ions are detected at the MCP plates. Then you can see that there is a shift in the peak towards the right side, and also the width of the peak is also gets broadened up, okay. So the peak shift is due to the increase in the average distance between pairs. Okay, this is the peak shift. Next is the broadening or widening is caused by the fraction of ions or atoms are removed, due to which there will be effect in the solute nearest neighbor analysis or the frequency distribution. Correct? Now the next thing is how detection efficiency will affect the cluster characterization. Okay. First we talked about the nearest neighbor. The second is how it will affect the cluster characterization. So the number of clusters, whatever the identified in an APT data set, okay, will be less than the number that physically existed in the original specimen. So if you have a real sample where there are certain clusters which are present, correct? So if these number of clusters are X, so now we will talk about the effect of detector efficiency on cluster characterization. Okay, so if you have in an needle specimen, if you have several clusters, okay, and if there are X number of clusters and which are present actually in your specimen, okay, but after the field evaporation, due to the limited detection efficiency, the number of clusters will be smaller than X number of clusters. Okay, so the clusters that are identified within the APT data only contain a also fraction of atoms, not all of them. Okay, so for large clusters, if you have a large size clusters, then the limited detected efficiency reduces the measured frequency of clusters. So it will reduce the measured frequency of clusters. So here you can see that there is a plot here where the simulated plot where you can see that by decreasing the detection efficiency, actually you are reducing the size of the clusters. Correct? The blue color is 100%, red is 57%, and green is 37%. Okay, so it also reduces the number of the frequency of these clusters. This is the effect of detector efficiency. Okay. Okay. So now we will talk about, again we will go to the radial distribution. So radial distribution, last class also I introduced it, so we will go a little bit into detail. Radial distribution is to identify the structure in a material system. Okay, how to identify? This is by examining average local neighborhood as a function of distance, okay, as a function of distance extended radially outwards from each atom in the data set. Okay, so this is used, radial distribution is also used to identify the solute or dopant short range order. in the system. Okay, so remember, if you have a series of atoms which have been detected in a data set, then radial distribution examines the average local neighborhood as a function of distance which is extended towards radially from a particular atom, and this is calculated for each atom position. Okay, and they gives the radial distribution map. Okay, so here the volume around each atom is divided into series of shells, or we can call it the concentric shells. Okay, so each shell is centered around an increasing large radius r and has a thickness of delta r. So here this is an image where you can see that these are the atomic positions. Okay, and if you see this particular atom at a radius r radially, and you can see that these are the concentric shells which is a thickness of dr. Okay, so each shell is centered around an increasing large radius r and has a thickness delta r. Now this algorithm, this radial distribution algorithm, builds a histogram of number of atoms in each of these shells. Okay, and this algorithm repeats the process centering each atom. Okay, so this is the histogram which plots the number of atoms in each shells, and this process is repeated at each atom, for each atom. Okay, every atom on the data set. So what we call is a RDF, which is given by 1 by rho NRDF R divided by 4 by 3 pi R plus delta R by 2 cube minus R minus delta R by 2 cube. Okay, so this particular value, denominator, is the volume of each shell. Okay, and rho is the average atomic density of the data set, and NRDF is the number of atoms in the shell at a distance r around each atom. Okay, so this is the function where, and this algorithm calculates this RDF at from each atom of the data set. Okay, so based on this, there is another term which is we call as a partial radial distribution. What is partial? So it nothing but it is a PRDF, which is nothing but a average chemical distribution. Okay, so what does it mean, average chemical distribution? It is nothing but the RDF analysis confined to the analysis of distribution of specific element in the system, not all the elements. Only if it is a specific to some single element, then these are called partial RDFs. Normal, if there is a RDF, you can consider that you are calculating for each atom, but if you are calculating for a specific element, these are called partial RDF. Okay, so it means that these are combined distribution, you can call it as a combined distribution of element A and B with reference to both A and B. Okay, this is the combined distribution of elements A and B with reference to both A and B. Now, another terminology is pair correlation. This is also a type of radial distribution for RDF analysis. This pair correlation is more specific. Okay, so this characterizes the distribution of atoms of a particular element, say B, or a group of elements B and C, like that group of elements, in the vicinity of atoms of a different element, say A. Okay, so this is given by 1 by rho, similar formula, NAB divided by 4 by 3 pi. This is the same term which is a volume of that particular shell. Okay, so this RDF can be used as a partial RDF, partial radial distribution, or you can also use as a pair correlation function. So here the rho B is the density of B atoms, and NAB is the average number of B atoms in the shell at a distance r around an A atom. Okay, it means these have a certain meanings. So if your GAB R is 1 for all values of R, it indicates that there is no correlation by the occurrence of that particular element. If your GABR is greater than 1, it means that there are high number of B atoms than the expected at R, at a distance R from the around the A atom. Okay, so this is the GAB. So now with this introduction to the radial distribution map, we will, so this is a one dimension, correct? So we are talking about in the one dimension. Now similarly, as the radial distribution maps, we have a spatial distribution maps. Okay, so this is spatial distribution map is nothing but a modified three-dimensional RDF. It means that it examines the average distribution of atoms in a particular crystallographic direction. In a particular crystallographic direction. Okay, so these are called spatial distribution maps. Now these spatial distribution maps, if you have a, can be done either in one dimension or they can be analyzed in the two dimensions. So if you have a needle specimen and this is your Z axis, you can generate these spatial distribution maps along this particular Z axis, which is a one dimension. Okay, and if you are generating a spatial distribution maps in X and Y, then these distribution maps are called two-dimensional spatial distribution maps. Okay, so we call it as a SDM. Usually these SDMs are very helpful in characterization of atomic planes in depth, okay, through reconstruction. So these atomic planes means you can actually calibrate DHKL value along the Z direction by using these SDMs. Okay, so I will give you some brief examples of this utilization of SDM. So you can see that this is a desorption map, okay, and you can see that these zones which are appearing on the desorption maps. As I told you, these zones are nothing but the crystallographic poles, similar to in the stereographic projection. Now you can take this particular region or this region or this region, and if you plot, you can actually by using SDMs, you can actually get the lattice planes of that particular pole. So here we can see that these are called 002 planes, which can be resolved, and these are the distance between these two lines is the interatomic distance, interplanar spacing of that 002. Okay, and if you plot these particular region in the data set, actually you can get these low density zones of each pole, which can be indexed for crystallographic poles. Okay, similarly you can also reconstruct the atomic planes of 113 planes, 111 planes. Okay, so these are the reconstruction in a one dimension. Now in two dimensions, actually you can also get the two-dimensional spots. Those spots are actually corresponds similar to the diffraction patterns what you are getting it, correct? So you can see that if you so this is in the Z direction, whatever you have plotted. So if you have a desorption map, you have a needle specimen. If you take the SDM along this particular direction and reconstruct it, you can actually do the you can actually image the atomic planes. But if you do in two dimension, in X and Y, actually you can get the spots which corresponds to the similar to the diffraction spots, correct? Now you can see that with this, if you plot, so these are the planes, atomic planes, and if you take SDM across these planes, you will get a sharp peaks at a certain defined interval, and the distance between these two is nothing but the D spacing, DHKL of that particular 002 plane, correct? You can see that this is the 1 bar 5 plane. Similarly, if you plot, this is in the Z direction, only one direction, correct, one dimensional. If you take in two dimensions, then actually you can see these spots. These you can similarly you can see as a diffraction patterns, correct? So this way these SDMs are very useful. Now I will give you a very brief example how these SDM maps are helpful to locate the site occupancy. Now here you can see there is an EBSD map, and the color on the EBSD map is nothing but these are the differently oriented grains. Okay, and the red color corresponds to near to 001 direction, green color corresponds to near to 101, dark blue corresponds to near to 111. Now if we identify a grain which is around 001, okay, so as I told you in Fib we have seen that how to make a atom probe samples. Now if I make a sample from this red region, and if you see the reconstruction and the desorption map, you can see this exactly is a four-fold symmetry, and this shows that this is along the 001 pole direction. You can see that, correct? And these are the precipitates which are present in a pure copper. Now this particular region, if I take the SDM maps along Z direction, and if you reconstruct along that particular pole, then actually you can get the 001 planes. We can see these golden color atoms which is having a constant distance of d spacing, which is corresponds to the interplanar spacing of the 001, and these red color corresponds to cobalt atoms. And with this resolution along the Z, actually you can get the composition of each plane. Okay, so this is how powerful it is, these SDM maps, which we can use for reconstruct the atomic planes and get the composition of that atomic planes. Okay, so one more example. It's a very to understand the site occupancy. So you know that in any unit cell, if there is a solid solution, the atoms can be randomly arranged. But if it is an ordered structure, so atom has a certain lattice points or lattice positions. Okay, so by generation of spatial distribution maps, as I told you, these are nothing but the average distribution of atoms in a particular crystallographic direction. Okay, and for each atom it is getting measured. Now here you can see that there is a unit cell which is a L12 ordered structure, and this unit cell has a two lattice positions. One is 0 0 0, another is half 0. These are the two lattice positions, and at these two lattice positions you have a different atoms. At 0 0 there are titanium atoms, at half 0 there are cobalt atoms. You can see here. So at this particular plane, if you see in this direction, this is called a mixed plane. Why mixed plane? Because it has both titanium and also the cobalt atom. But if you see this particular plane, it is a pure plane. Okay, pure plane means only cobalt atoms, and here it is a titanium plus cobalt atoms. Now if you take the distance between these of a unit cell, it is 0.36, and this pure plane is at the 0.18 nanometers at the center of this 0.36. Now I will do the reconstruction or the atom probe, and you can see that these are the lattice planes. Fine. Now if you plot a SDM map along this particular direction for this particular unit cell, you can see that this is the distance 0, 0.4, 0.8, and you can see that at each 0.36, this is at 0.36, this is at 0.72, you can see that the titanium peak appears. Okay, it means that the titanium atoms are located on 0 0 0 positions, which is at a distance of 0.36. But if you see for the cobalt distribution, you can see that the each peak appears at 0.18, 0.36, 0.54, 0.72. So because one peak is mixed and one peak is pure, so you will get presence of cobalt atoms at each 0.18 distance. So from here, from these SDMs, what you can find out is the titanium is occupying the 0 0 0 positions, while the cobalt occupies the half 0 positions. So this is how these spatial distribution maps can be used to identify the site occupancy of any unit cell. If it is an ordered structure, you will have a different elements sitting at different atomic positions. If it is an ordered structure, if it is a solid solution, what you will see is you will get a titanium peak even at 0.18 also, 0.54. So you will get a solid solution, because the titanium can occupy any of these either 0 0 positions or half 0 positions. Okay. Okay. So with this, in this class, what we have taught is, we have taught about the effect of detector efficiency. Then we talked about the radial distribution maps. radial distribution, RDF analysis, radial distribution for frequency. Then this radial distribution frequency is along the radially, correct? And if you want a 3D representation of this, we have a spatial distribution maps, which are very useful to find out what type of atom is present at what type of site of the unit cell. Okay, so with this I will end this class today, and what we do is, now most of the atom probe topics have been covered now. So now in the next class I will start with the basic electron microscopy, and after covering the electron microscopy, it is not only the transmission, so it will include transmission electron microscopy and also the electron channeling contrast imaging and SEM. And I will show you how these three techniques, in the last few classes, maybe 2 or 3 classes, I will go through the application of these techniques in a correlative way. So we will cover some correlative microscopy by giving certain examples, real examples, and how these microscopy techniques can be used to identify the mechanisms or the problems in the material. Okay, thank you.